science friends, I'm going to try to keep this lesson pretty short today, but it is very important because we're going to use this information when we do a virtual lab that we're going to work on Monday. It's also important because we're talking about a concept that's easily confused with yesterday's concept. So yesterday we talked about sex-linked genes. Today we are talking about linked genes, which are different from sex-linked genes. Sex-linked genes indicate genes that are on a sex chromosome, so an X or a Y chromosome. Linked genes, are genes that are located on the same chromosome that tend to be inherited together, okay? So generally, these linked genes are close together on the same chromosome, and you want to write that down, okay? The closer together, probably want to make a note of this, the more likely they are to be inherited together. And so the further apart, the less likely they are to be inherited together. And I'll explain why that is a little bit later. But for now, that would be a good thing to probably add into your notes here. Okay, so linked genes, doesn't matter if it's on the sex chromosome or a different chromosome, but they're genes located on the same chromosome and they tend to be inherited together. Okay, so this is a continuation of Thomas Hunt Morgan's study. Remember, we talked about Thomas Hunt Morgan studying the fruit flies the other day, the University of Kentucky graduate. So in the fruit flies, in the Drosophila, he took this fly here that was the wild type. Remember, the wild type is the normal phenotype. It's what you tend to see more often than not. And so the wild type has a gray body and normal wings. So here's our gray body. These are normal rings, meaning they're long. They're the normal size, okay? Now, the other option, if it's not a wild type, it's considered to be a mutant. And this one is a double mutant, meaning it has both unusual sets of characteristics. So it has the black body and it has the vestigial wings. So these are vestigial wings that are a smaller wing, okay? So if we look at the genotypes for these, we have the pluses are going to indicate the wild type, and then the ones without the pluses are going to be the mutant. Okay, so when he crossed that wild type and that double mutant, everything that he got looked like this. And that's exactly what we would expect if we put our Punnett square together. B plus B, VG plus VG. So we got 100% of those, okay? Just like we would expect. And it has the wild type phenotype, but technically we know that it's going to be heterozygous, so to speak, as far as its alleles are concerned. So when he took a female of those and crossed it with a male of the double mutant, okay? If we put this in a Punnett square, this is what we would get. This double mutant male is always going to give little b, little vg for its sperm. And so we see that here, okay? The female can give b plus, vg plus. It can give b plus, vg. It can give B, VG plus, and it can give B, VG in the eggs. And that's what is documented right here, okay? So normally, if we were to work out this Punnett square, I just foiled those, essentially what I did here, we would expect a 1-to-1-to-1-to-1 ratio, right? 25% of each of these phenotypes. That's what we would expect if these traits were located on different chromosomes, okay? But that's not what Thomas Hunt Morgan saw. I don't know the exact numbers off the top of my head, but it's something like about 900-ish of each of these and about 200 of each of these, okay? So what he saw was a lot of this wild type, a lot of the double mutant, and fewer of these other two, okay? Now, these two are what we call parental traits. They have the phenotypes of the parents, okay? Or actually maybe of these parents. These parents are really the ones I should have drawn my arrow to. They have the phenotype of their parents, and so we call that a parental phenotype. These others over here are what we call recombinant phenotypes, okay? And so because we saw different ratios, or I'll say we, I really mean Thomas Hunt Morgan, than we expected, then that's indicating to him that there's something else going on here, okay? These chromosomes or these genes are not located on different chromosomes. If they were on different chromosomes, we would have expected this 25, 25, 25, 25 ratio, but they're not. So that means that these genes are likely located on the same chromosome, and those parental alleles are frequently inherited together, okay? So what Thomas Hunt Morgan studied is known as genetic recombination, okay? He looked at the production of offsprings with combinations of trait that were different than what was found in either parent, okay? Just like we saw here with these last two options. They demonstrated genetic recombination, okay? So we have our parental phenotypes and we have our recombinant phenotypes. Mendel saw those things happen too. He just didn't study it and understand it as well as Thomas Hunt Morgan did, okay? So for Mendel, let's look at a Mendelian example. He had the yellow round peas with this parent, okay? So if we FOIL this, big Y, big R, big Y, little r, and little y, big R, this is the alleles that parent would produce, right? And the other parent, green and wrinkled, would always give a little y, little r. So again, if we did this Punnett square, we should expect a 1 to 1 to 1 to 1 ratio, or 25% of each, okay? These being the parental offspring, meaning those were yellow and round, just like the mom, and these being green and wrinkled, just like the dad, okay? These other two are the recombinant offsprings. So they have the yellow from their mom and the wrinkled from their dad, or the round from their mom and the green from their dad, okay? That's a recombinant phenotype. A different combination of the phenotype from both parents, okay? So what causes that? Well, recombination is caused by crossing over. Remember, crossing over happens during meiosis. It happens during prophase 1 of meiosis. Crossing over is when you have two sets of chromosomes, homologous chromosomes, one from each parent. So this is mom and this is dad. And they synapse or come together. And those two non-sister chromatids exchange, right? Those two non-sister chromatids exchange. And so now, if this gamete is going to get mostly dad, but a little bit of mom, okay? And so that's where we get recombination. So scientists are able to now look at genetic maps. So what genetic maps are is they look at a chromosome and they look at what genes are on a chromosome. You've probably all heard of the Human Genome Project, where they map the human genome and say at this spot, this codes for this. And so that's what a genetic map is. It shows the loci on a chromosome and what that segment codes for, okay? And so they can use these genetic maps and use recombination data to make what is known as a linkage map. And this is a linkage map down here, okay? So I told you up here at the beginning that if you have genes that are close together, they're more likely to be inherited together, okay? So let's look at this chromosome. Let's say we have a gene for eye color. Now I'm making this a lot more simple than it is, but let's just hear me out. And height. And then we've got one for hair color here. Okay. Now, if you're going to have crossing over, what is the chance that height and eye color will get separated by a crossover event? Probably a pretty good chance there, right? Because they're very far apart from one another on the chromosome. So if we know that this chromosome is going to be split, and two pieces of it are going to be inherited separately, it's like it's probably going to happen between eye color and height just because they're so far apart, right? Versus eye color and hair color are closer together, so it's less likely to be split because they're close together. Okay, let me give you a different example. Let's say we're going to look at this at the school in your 12th grade or in your 11th grade class, in the junior class, and we're going to divide it up by alphabet. Okay, so my last name is Brown. So my name is Brown, and Mariah, she's in our class, so I'm going to use her as an example. Her name is Bolin. So if I'm in the same grade as Mariah, we're probably going to be in the same group because our names are close together. Okay, versus Mariah is probably not going to be in the same group with John David Rains because John's last name is Rains, and that's really far from Bolin, right? So it's just because those are far apart and we know they're going to be split up into groups, it's more apt that Mariah and John would end up in different groups than it is that me, Kelly Brown, and Mariah would end up in different groups because our names are close together. Does that make sense? I know you can't answer me, but hopefully it makes sense. Okay? So the further apart genes are on a chromosome, the less likely they are to be inherited together. That means they are more likely to be separated by crossing over. More likely to be separated by crossing over. If they're close together, they're more likely to be inherited together. They are less likely to be separated by crossing over. Okay? So hopefully that makes sense there. So generally what we say is if it has a recombination frequency greater than 50%, then they are on different chromosomes. If they are inherited separately more than 50% of the time, then they are on different chromosomes. If they have a recombination frequency of less than 50%, then they're more likely on the same chromosome. Okay? The smaller the recombination frequency, the closer they are together on the chromosome. Okay, that's really sloppy. The closer they are together on the chromosome with a smaller recombination frequency. So let's look here. We've got a recombination frequency shown here. These are also known as map units. 1% recombination frequency is 1% or is 1 map unit. And sometimes you may even hear them referred to, I don't think AP uses this term, but in the lab or in the field of biology, centimorgans, named after Thomas Hunt Morgan. So that means that these chromosomes will only recombine, mean not be inherited together, 9% of the time. Okay? Most of the time they're inherited together because they're close together. Versus this chromosome, the VG and the B, they are separated a little bit further apart. So if crossing over is going to happen, it would be more apt to happen between B and VG than it would between B and CN. Okay? And this diagram is a linkage map. Now, these distances, these centimorgans, they don't actually correlate to a distance like a centimeter, a millimeter, micrometer, or anything like that. So they just show location relative to one another, but not a specific location. So they show order, but not a specific spot. Okay, and that's important. All right, so I think that's got everything that we need to know about recombination and map units and all of that type of stuff, genetic recombination, gene frequency. If you have any questions about it, let me know. If not, then we will try to put this concept to practice on Monday with our lab. You all have a great day and a great weekend.